In this video, we will discuss about the impact of different properties of macromolecular crowder on the cell surface enzymatic activity and the mobility of enzyme on the cell surface. In the previous video, we have understood or we have seen the results where it is clear that with the use of macromolecular crowder, that is Dextran, in the previous video, we have seen that it will cause increase in the and cell surface enzymatic reactions by manifold. It reaches to a maximum of 440 fold with this enzyme BFGH 110A with dextran. So here they wanted to check the impact of different properties of macromolecular crowder. So first, and here they use different macromolecular crowder at different concentrations, and these concentrations can be related to the biopolymer concentrations found in the body. That is 50 milligram per ml uh, is is found in the esters. Not able to exactly recall it, but those information are given in the paper. This 100 milligram per ml is in the blood plasma, and 300 milligram per ml is in is within the cells. And this 50 milligram per ml, I think, is in the uh, extracellular region, but you can confirm this by referring to the paper. So they used different macromolecular crowders of different molecular weight. It is dextran at different molecular weights, phycol and HPG, and they see a low enzymatic activity at 50 milligram per ml and also at 100 milligram per ml. But for this 300 milligram per ml, they see a high enzymatic activity, uh, and those enzymatic activity is highest for the HPG at 30 and 110 kilodalton compared to the dextran and phycol. And after that, they compare the enzymatic activity of dextran 40 kilodalton and HPG 30 kilodalton at different macromolecular crowder concentrations, and they can find out that HPG at 30 kilodalton will give more enzymatic reactions compared to the dextran at 40 kilodalton. So this is, but they do not see too much impact of the they do not see too much impact of the different molecular weight of the macromolecular compounds, except for this HPG, where they see high difference between 10 kilodalton and 30 and 110 kilodaltons. But for dextran, they do not see too much difference in different molecular weight compound, different molecular weight of the this macromolecular crowders. And after that, they wanted to check whether these macromolecular crowders affect the mobility of the enzyme on the cell surface. So for this, they use the FRET technology, where first they immobilize the cells on the cell surface and then photo bleach it using the fluorescence. And once they photo bleach, and they again check the fluorescence, and in this case, if they see the fluorescence, this could be because of some of the enzymes, some of these macromolecular crowders diffuse inside the layer that has been used, and those enzymes will give us this green fluorescence image. So initially they check it for the 40 kilodaltons at 90 milligram per ml and 320 milligram per ml, and they can clearly see the increase in the fluorescence signals, indicating that the high concentrations of these dextrans are expressed on the cell surface. But I want to confirm that these results, and we will go on that topic later on, but first we will understand this panel B. Panel B shows about the different concentrations of the dextran 40 kilodaltons and its normalized fluorescence intensity compared to the PBS. And what they have able to find out is that at maximum they are able to see 5.5 fold increase at 320 milligram per ml 40 kilodalton dextrans compared to the surface with only PBS. Next, they wanted to check whether these signals are from the diffusions of this diffusions of the enzymes within this immobilized surface layer or not. So for this, what they did is uh, they photo bleach the cells uh, once they are coated on the cell surface. They photo bleach the once they are coated on the surface. They photo bleach the cells, and then after some time they again check the fluorescence intensity. And in that case, if some of the cells are immobilized before photo bleaching, they will give us some signals. And what they are able to find out that in the PBS they see most in the case of PBS they see a a decrease in the case of PBS they see a increase in the fluorescence signals with time, but for the Dextran at different concentrations, that is 40 kilodaltons at 90 milligram per ml, 180 milligram per ml, and 320 milligram per ml, they see a low increase or minimum increase in the fluorescence intensity after initial time period. So this indicates that the maximum diffusions occurs in the PBS, the minimum diffusions or diffusions lower down to some fold in the case of macromolecular compound, it is 40 kilodaltons with 320 milligram per mg, 320 milligram per ml. And these things are quantified in the panel D, where we can see that they did their experiment for the bulk surface and the cell surface. For the bulk surface, they see the bulk surface, they are able to see like diffusion coefficient of around uh, 30 32 kilodaltons for the cell surface and 45 kilodaltons for the bulk surface. And this goes down, this diffusion coefficient goes down with the increase in the polymer concentrations, and it goes down to like around 2 to 3 micrometer square per second for the cell surface, and around 15 micrometer square per second for the bulk surface. This indicates that with increase in the macromolecular concentrations, the diffusion coefficient or the diffusion ability of the enzymes goes down, indicates that cell surface, these enzymes will remain, the enzymatic reactions will occur on the cell surface, not within the cells. Or these enzymatic reactions occurs, most of the enzymatic reactions will occur on the cell surface, and very minimum of the enzymes might occur within the cells. This is what their experimental result shows about the effect of different properties of macromolecular router on cell surface and the mobility of enzymes on the cell surface. If you need to understand more about these experiments, and they also have some results in the supplemental figures, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.